Hi, this is Ms. Klemme, and welcome to the podcast on protein synthesis and mitosis in cell division. Now, before I begin in the process of the topic of this PowerPoint, I want to introduce you guys to two very famous scientists by the name of Watson and Crick, and they're pictured here. They're famous for deciphering the structure of DNA in the early 1900s. And they studied it, but their bodies are no different from ours. Their bodies are made of trillions of cells, seen here, and those cells are controlled by DNA. Now let's flip this equation here. If we go from DNA is the controller of all of our cells, and our cells, based on levels of organization, make up us. However, I'm not telling you the whole picture. There's actually a missing link between DNA and cells right here. And that missing link actually is protein. Now, proteins really do make our cells. They make our hair and our nails and our teeth and our organs and our muscles. All of that stuff is protein. All of those things are made of protein. So essentially what DNA is building, is coding for, is not our cells, it's instructions on how to make proteins. And so those proteins then can go on to be organized in such a way to make different types of cells, and those cells make us. So keep that in mind. Sure, DNA does get all the glory, we uncovered its structure, but proteins are the ones that do all the work for us. Now, there are two steps in making a protein. But there's a problem we have to overcome first. So say this whole slide right here in front of you is a cell, and that little oval inside is the nucleus. And we know inside the nucleus is DNA. And we're going to say that that DNA is locked in the vault. The vault being the nucleus. It can't leave. And it can't leave because it's so fragile and precious, we don't want it to get messed up or anything if it would leave into the cytoplasm. Here's the problem. We know that ribosomes are the makers of proteins, and those ribosomes, they hang out in the cytoplasm. So in order to get the instructions to make proteins from the DNA, we've got to get the DNA to the ribosomes. So here's a solution. So we know our DNA is locked in the vault. So why don't we make a copy of it? Not an actual copy with DNA, but let's use something really similar to DNA that can leave the nucleus. So then it can leave the nucleus, go to the ribosome, and make our proteins. Well, that copy that we make is actually just a messenger. In fact, that little lowercase m in front of the RNA is called messenger RNA. It literally carries the message from the DNA to the ribosome. So there's your solution. Now that first step is called transcription, and I'll show you how it's done right here. Okay, so say this is a small little chain of DNA with all those bases, those A's, C's, T's, and G's. Now if we were just making another strand of DNA, every time you see an A it would pair with a T, and every time you see a C it would pair with a G, and vice versa. However, if we want to make a very similar but not an exact copy, when we want to make messenger RNA, those bases change a little bit. So instead of pairing A with T, A pairs with a U. It's a base called uracil. So let's make our matches. If this is DNA, A will pair with U, C still sticks with G, AU. T can still pair with A as long as it's not a T, and G with C, T with A. So there's our messenger RNA. This will leave the nucleus and it's going to go to the ribosome. And that's all transcription is. It's making a similar copy of DNA that is able to leave the nucleus and head for the ribosome. Now, here's our messenger RNA with all the letters. Here's our protein chain of amino acids. Now we know that this happens at the ribosome because the ribosome puts together these amino acids into proteins, but how does it do that? Well, let's take a look. Let's recap what we have so far. We have the DNA, it's locked in the nucleus in the vault, and it undergoes transcription. It makes a similar copy in the form of mRNA, which is right down here. The second step and final step to building a protein is called translation. So it's basically taking the language of A's and C's and G's and U's and converting them into an entirely different language. It's translating it. And the ribosome is the one that does the translating. The ribosome is bilingual. It can read RNA and it can read protein. And so it reads the RNA, and from that it translates it into a different code, into an amino acid code. And those amino acids are put in order in such a way to make a particular trait in your body, to make the facial features that you see, how you look or how you metabolize something. Those are all traits controlled by proteins. And proteins are made from the instructions in DNA. So make sure you know those two steps in protein synthesis. So, a little bit of a recap here. In an earlier podcast, we talked about cells making energy, and we talked about cells making proteins. The currency of energy is ATP. That's what the cell is making, and it's making that in the mitochondria. And to make proteins, we just talked about that. Cells go through transcription and translation to do this. The final task of a cell is to make more cells for growing and repairing. And there are two ways that cells make more cells. The first way is mitosis, and then the second way is meiosis. So that should be an E. We'll talk about mitosis first. Okay, so in mitosis, two things have to get divvied up from the parent cell to make two daughter cells. Because remember, we're really starting with one cell and we're dividing it in half equally to make two. So what gets passed on is an exact copy of DNA, the genetic material. And that process is done through mitosis. However, the cytoplasm with all the good organelles has to be divvied up as well. And that process usually happens right after mitosis. It's called cytokinesis. Now, what do these steps look like? Well, the first step is interphase, and that's just when the cell is growing, it's doing its thing. It's making the proteins that it was born to make. And you can see there the chromosomes are just like a ball of string. The next phase is prophase. You can see the chromosomes here are thickening. The centrioles right here are moving to the poles, to the outer edges, and they're releasing little ropes called spindle fibers. And as well, the nuclear membrane, the white coating around the DNA, is starting to erode. In the third phase, metaphase, you can see now the chromosomes have all lined up on the metaphase plate, the equator, and they're all attached to the spindle fibers. So that in anaphase, those spindle fibers can kind of like reeling in a fish, can reel in half the amount of chromosomes. Because, and I almost forgot this part, in interphase, the most important thing that happens for mitosis is the DNA replicates. It makes a copy of itself so that each new daughter cell can have the exact same set of chromosomes. And that you can see here happening in anaphase. The last phase is telophase, and it pretty much reverses what happens in prophase. The chromosomes start to decompress, the nuclear membrane starts to reform. And then cytokinesis happens pretty much at the same time, which is the division of the cytoplasm, so that you get two new cells. And that's mitosis. Some things that can happen in mitosis, some adjectives for it. We have some opposites here, hypertrophy and atrophy. Cells can increase or decrease in size depending on how cytokinesis occurs. Hyperplasia, which is when cells start to divide excessively more than they need to. Anaplasia, where they're not maturing properly. And both of those can combine to the ultimate, which is cancer, and that's an uncontrolled mitosis. The cells are not only reproducing at an excessive rate, but they're reproducing improperly with lots of errors and mutations. So to recap, mitosis produces cells that are exact same, they're identical clones, and they have the same number of chromosomes. If the parent has 4, the offspring daughter cells will both have 4. Now let me introduce you just really briefly to meiosis, which is another type of cell division, but we're only using it to make special cells, to make sperm or egg. And the main goal of meiosis is to cut the chromosome numbers in half. So if we're looking at a human cell, just a regular cell, they have 46 chromosomes. If those cells get cut in half with their chromosome count, they make a special type of cell called a gamete, which is a sperm or egg cell. And that process of cutting the chromosomes from 46 to 23 is known as meiosis. It makes a new cell, so it's a type of cell division, it just doesn't have the same amount of chromosomes. And what does this really mean for all of us? Well, here's why meiosis is important. We start with regular 46 chromosome cells, and if we cut them in half to make gametes, that's meiosis. Now the reason we have to cut them in half is because when they unite again for the process of fertilization, then our baby has the right amount of chromosomes. It has 46 like it's supposed to. If it didn't cut the chromosome number in half, our baby would have way too many chromosomes, and that would not be viable. Now once that baby has 46 chromosomes, it starts to divide and grow by mitosis until we eventually get a full-sized newborn. Okay, so meiosis is used to make those special cells with half the number of chromosomes, and then once that baby is made through fertilization, mitosis is used to grow in size. To recap meiosis actually produces cells with different information, because those chromosomes that we see, they can actually exchange information and swap genes. So you get unique combinations of daughter cells, not exact copies. And the number of chromosomes